Hi, I'm Marian Smallin. I am a shared grad student at Moffitt Cancer Center between Dr. Andrew Brohl and Dr. Sherry Phelan-Thomas. And I'm here at SITC presenting my work on how a cluster of human microRNAs, when overexpressed, can impact immunotherapy in cancer. So we, in my lab, investigate 46 human microRNAs that are all expressed at once. And we found that this actually can have implications in cancer that can affect treatment decisions. And so one of the things that we had to do in order to study this properly is we had to create these CRISPR models that are very unique in the sense that they overexpress all 46 at one time, whereas usually in the past, the only models that have existed have expressed one. So once we made those, we started to investigate how the tumor microenvironment shifts in response to the overexpression. And so we did some co-culturing experiments where we identified that there was a less effective killing mechanism of our T cells in comparison to our controls. We then ended up doing some more deep diving, and we found out that it's because of the expression of HLA molecules on the surface. They actually get repressed significantly, and this is not an interferon gamma dependent mechanism. So we also then investigated antigen presenting cells, and we found that similarly on the surface of those antigen presenting cells, HLA class 1 is affected, but not class 2. And we've been able to trace the class 1 changes to overexpression back to the core transcription factor that navigates them. And so now we are investigating how our microRNAs directly bind this transcription factor in order to figure out the mechanistic implications of what's going on. So this is a very mechanistic project, but we're eventually hoping to transition to play around or research the possibility of developing an RNA therapeutic that can target all 46 of these microRNAs at the same time in order to use it as some sort of adjuvant in addition to immunotherapy.